Good morning, good afternoon, or good evening, wherever you are in the world. Thank you very much for taking time out of your schedule to join us for this Pistoia Alliance webinar on data quality for LLMs, building a reliable data foundation. My name is Sahid Tharia. I'm a consultant at the Pistoia Alliance, and I'm absolutely delighted that we have Abhishek Jha, the co-founder and CEO of Elucidata, who's going to be speaking for us today. Previously, AJ was an early team member at Agios Pharmaceuticals, where he brought 3 first-in-class drugs for AML patients to the market. And his experience at Agios inspired him to start a business with the mission to give back every possible hour to the scientists like him, so that drugs can get to the patient as quickly as possible. So, Abhishek, welcome.
Thanks, Zahid. And I also wanted to thank all of you who are participating. I understand everyone has a busy schedule, so thanks for taking time out. I also want to thank Pistoia for being a wonderful community for folks like us, at least organizations like us, to share and talk and advance a conversation around this data at large. LLMs for the last 15 months has generated a lot of interest, also in our industry, and we wanted to take this opportunity to again share notes of things that we have learned of about working on this for many months now with a very strong team. There's some things that are that we are ready to publish, something that we cannot go into a lot of detail, but hopefully I think we'll have a very good conversation, and hopefully we can advance the understanding and appreciation of the challenges and the opportunities of what LLMs mean for the industry at large. Just to set the context, right? We all have benefited from this. Our careers have benefited, and our outcomes of this growth in data. We have contributed to the growth. We have used the data. So if you look at this funnel, sideways funnels, on the left you have the entire universe of data, which, to be honest, is very poorly characterized. We don't know how much of the data is being generated from what source, where is it. The quantitative metrics of this universe is very, very hard to capture. But qualitatively, we all agree and know that there are more types of data that are coming up. There's more volume of data that is coming up, and arguably also at an increasing velocity. Now, if you move to the other end of this funnel, which is where most of us operate, we have made a schematic, a cartoon, if you will, of what the current state is, which allows you to use data, but only a very small fraction of it. So think of any small or useful tool that you may use quite often, making a Disney plot or PCA, or even a ML model that you might run. All of those things tap into a very small section of the data that is available, and that's for a variety of reasons. But I'll not go into those reasons just yet. But what we can agree upon is that a very small slice of data that is available at large is being currently fed into your R&D pipeline. Now, one of the excitement about Gen AI and LLM and tools like that is that it promises to push this further to the left, where a bigger swath of data, a bigger subsection of data would be increasingly and easily available for you to use, right? To derive decisions, to drive your research and development programs. And to highlight this example, I want to give a shout out to a paper that we have been enjoying and reading a lot, a single cell GPT paper that was in bioRxiv from Bo Wang's lab in Toronto. And as if I remember right, it has been recently accepted in Nature Methods too. Now, this is a plot that I have borrowed from the paper. And what it shows is that, on the x-axis is the number of cells. So if I go back to my last slide, it talks about the amount of data that is available to this model, the single cell GPT model. And as you move from 30K to 33 million, you see that the performance, which is captured on the y-axis in terms of accuracy, improves, right? Now, there are very few tools, and single cell GPT is one promising such tools, which allows you to access and use and leverage more data, in this case, around 33 million cells, where this single cell GPT model was trained on with real value, because the performance, the accuracy improves. Now, is 33 million enough? Why not 330 million? And is the value, the accuracy, or the tasks that single cell GPT performs? We will not get into all of that. But what I wanted to highlight is the tools like these single cell GPT will become more available, which allows groups, individual scientists and groups, to access more data to drive their R&D program in a meaningful way. And this is just one example. There are many other papers that have highlighted this as well. AlphaFold is yet another example of how a lot of data of resolved protein structures has been very, very helpful in determining structure, predicting structure of other sequences as well. So if we were to synthesize and condense a lot of things that we are seeing, what we are observing in across different groups in life sciences, mostly in early stage R&D, or also in later stages for big companies and small companies, also some academic labs and nonprofits. There are a few things that are emerging quite clearly, and we are very keen and willing to hang our hat on that. Number one, which is less of a it's kind of given, or it has been widely accepted and embraced, is that multimodal data is of value. It was not quite obvious even 5 to 10 years back, but increasingly that has become the default and mainstream, and we welcome that very, very. We are very, very excited about that shift. And in this case, specifically, we are talking about a whole slew of data, right? Like omics data from NGS platforms, like single cell, bulk, the usual suspects, right? We are also talking about omics data from mass spec platforms like metabolomics and proteomics, assays, RWE data, and so on and so forth. That, I think, is here to stay, and there's a critical mass of the industry which has embraced that and wants to execute on that. The second part that is also becoming clear is the foundational models, we believe, and it remains to be seen, will be very specific, focused on a particular task. For example, AlphaFold does protein structure prediction. It does not do like protein function prediction, for example. Or single cell GPT model, performs 3 or 4 tasks. One of them is cell type annotation. So you can imagine a whole slew of tasks, right? A whole catalog, if you will, to predict ADMET properties or rank the targets or match gene signatures. And we argue and believe that those specific task-specific models will become more commonplace. Either they will be published from academia or also in some from companies like DeepMind from Google. Irrespective, they will be available to the community to use and leverage that and advance their R&D programs. They may be not trivial and easy to use, as we already know from our experience of using publicly available data or publicly available models. Now, moving to the third piece, as to how you consume the data. So far, if I can split that into 2 categories, there's one which is more friendly to data scientists and computational biologists, where they interact with the data in a coding environment. For example, more API, SDK-based environments. The second is where biologists or chemists who are more closer to the data and less computationally savvy. They use GUI-based tools like Spotfire, Prism, just to name a couple from a list of many, to leverage data to extract insights. We don't believe that those two will go away, but what we are arguing and seeing that the third way to consume data will emerge as well, which is where folks will want to just share instructions in natural language, at this point mostly English, and ask questions to the data, much like the way they do to their colleagues. And we'll later in the presentation, I'll also highlight one case study, not in a lot of depth, but just to illustrate as to what could be possible from such consumption journeys. We believe and we argue that that will scale up the bioinformatics teams, which are typically small and even if they are large, they are definitely swamped. They're handling queries from all their colleagues, and some of those queries are quite repetitive and are very amenable to the advances in LLMs that have been made, and could really free them up so that they can focus on higher value problems at the same time, without compromising the support that their colleagues need. So, just to capture the three aspects of what we are seeing and what we're hoping this to be more mainstream in the next 12 to 18 months, is an embrace of multimodal data, very task-specific foundational models, and consumption journey that is enabled greatly by instructions in natural language. Now, to enable all of this, one of the things that we have learned and we have shown, I hope to highlight some of the results in today's presentation as well, is that high quality data will be critical, not nice to have, but critical to have for task specific models. Of course, I mean, there's a more nuanced conversation as to what high quality means and what tasks rely critically on it and what don't. And we have, an evolving understanding, and we'd be happy to dig deeper and have a more uh, in-depth conversation around that. But at a high level, qualitatively, what we see is that high quality data is essential. So if you imagine a pyramid of sorts, right? You have generic data that comes from the internet. Then you have contextual data. Again, if you think about it, right, a random paragraph from New York Times does not, or Wikipedia, right, does not read or feel like a paragraph from a paper in PNAS or in bioarchive, right? And that context is important to capture for any foundational model. So on top of what we are calling generic data from the internet, table, text, we would need to put a layer of contextual data from arguably from the public domain, because the volume is immense and it's valuable. So why not, right? And at the very top, we think highly curated, very high quality harmonized data would be needed to perform that specific task. So, for example, if you have gene signature from RNA-seq data, right, you'd want a corpus of RNA-seq data to be harmonized to the highest extent possible. And we have, gotten into specifics of what that means in separate forums, in white papers and blogs and other webinars as well. Again, happy to answer questions, but I'll not go into a lot of details about that just in today's presentation. So we think that will be the third topmost layer, perhaps less in volume, most definitely less in volume, but very high in quality. And this would be the construct of what would underlie any task-specific model. So let's look at a very small, quick example of what that means for a specific task. And in this case, the task is what is called NER, a named entity recognition. So if you look at the sentences on the right, you can imagine these sentences come from a paper, from a clinical note, or a variety of sources. And the task here is for the model to name the entity, in this case, disease, cell type, and tissue. And as a human expert, you're quite wired to, after years of training, to pick them up quite nicely. Increasingly, what we're seeing is the models can also capture that quite effectively. And here's some result that we are sharing, where we have defined this task for 4 entities: cell type, tissue, cell line, and disease, mostly from abstracts from PubMed. And the 3 bars that capture the performance, in this case by quantifying F1 scores, which capture both the precision and the recall. And you see that there's a clear trend, right, to different extent. But irrespective, it's very clear that harmonized data, when you have that topmost layer in the pyramid I was showing you earlier, outperforms the other 2 bars, right? And when you have the most generic data alone, the performance is quite, modest. And you improve that by adding more contextual data, and you get even more improvement and advance um, uh, in performance if you have harmonized data as well. Now, the red line that you see is what we are calling the ground truth, where human experts go in and look at the same biomedical abstracts and call out the same entities, in this case, tissue, disease, strain, and more. Now, human experts can also be wrong at times. We all have we all know that we have made such mistakes as well. But irrespective, for the purpose of this conversation, we'll treat that as the source of ground truth, what we call expert curated data. So, even in a small, simple task like this, we see that harmonized data, having a layer of that on top of it, increases the performance. For some tasks, this improvement is quite massive and critical. For others, it's quite modest. Again, that is what I think requires a more deeper conversation. It's not quite amenable to a 30-minute, 40-minute webinar, but we think and talk and work a lot on this, so we'll be happy to um, have any dialogue around that. Now, if we buy the premise that high quality data is indeed a critical piece of um, building models like these, you need to have a platform, a engine of sorts, to harmonize data at scale, because a lot of the foundation models, and increasingly we are seeing that companies, commercial companies, biotech companies are trying to make foundational models as well, would rely on huge volumes of data. Not all of them would need to be at the same level of quality of curation or harmonization, but a good chunk of it, the top layer of it, would need to be able to do that. And for that, we have been, for the last many years, we have been able to build a very scalable harmonization engine, which works on the different types of data to give a very high quality data at the end of it. And there are three elements to this engine. Number one is the technology, which is powered by LLMs. And we as a group were quite well placed, because earlier we were using BERT, which was published by Google, and more recently we are using other LLMs as well in addition to BERT. So that's one piece that gives us a huge advantage of speed. We also have human curators. These are trained bioinformatics scientists, NLP engineers, data engineers, who support the curation effort and are in the loop to evaluate the model of the performance, to build the models to begin with, to fine-tune the models. And the third pillar is the process. For example, we give out the same data to two curators blinded. If we train the curators, we track the performance of the curators, we intervene when the curators are not performing as well. So there's a lot of process inbuilt to ensure the success and the viability of the scalable harmonization engine. And this is working quite reliably and robustly in our hands, and is creating value with partners that we have been working with. For example, just to give you some sense of scale, um, we have been able to deploy our harmonization engine on more than two types of data, omics and non-omics as well. At this point, mostly focused on text and tabular data, but we're also including images in our roadmap. Cumulatively, we have harmonized around more than 50 million samples over the last 5 years. On a monthly basis, we harmonize around 35 terabytes of data, and we have a buffer to expand that by 3 to 4x. And a lot of this is built on ETL pipelines and harmonization engine that we have built over the years and months, with a very strong team that is continuously focusing just on defining this and refining this harmonization engine. So I hope this sets the stage and gives a background of what we do as a company, how this fits into this broader conversation around LLMs that we as a community are having almost on a daily basis. There are papers that are coming out, there are companies that are getting formed. With that, context and background, I want to dive deeper into a benchmarking study that we have done, which essentially, again, for a lot of us in the room, it is kind of obvious, not for all, but for most, that high quality data would increase performance. But to our knowledge, no one has quite systematically evaluated as to what would high quality mean, and how significant would the performance improvement would be. And we have in the last 3-4 months we have been able to do a very systematic study, which we are also putting out in the public domain with a in a white paper form. Hopefully we can translate that into a bioarchive as well, that we can put out for the community to use and comment and have a conversation around. But today, in the next 10-15 minutes, I'll walk you through the benchmarking study that we have done. So here's the experimental design. So let's get oriented about that for a second. What we did was we went to a published corpus of data, which is CREEDS from Avi Mayan's lab. And there are some numbers to capture the extent, depth, and breadth of this corpus of data. Mostly data from geo, around 30,000 samples, many diseases, hundreds of perturbations, and also drugs, right? Now, CREEDS, to our knowledge, is the most recent and most expansive corpus data that is available in the public domain. It came out around 7-8 years back, but we did not find anything. And if you guys know, please let us know. We'd be happy to compare against that too. CREEDS, to our knowledge, is the most comprehensive and largest and most recent corpus that is available for such comparisons. And perhaps that's for a reason, because it's very hard to curate even 30,000 samples. And Avi Mayan lab did a phenomenal job to do that and also make it available to the community. So that's one arm of our experiment. The other arm is we take the same data sets that are in CREEDS, and we go to the parental source, in this case, geo. And that becomes the baseline of sorts, right? That's data at source. And finally, the third arm of the experiment is what we're calling polyharmonized data, where we take the identical same 1950 data sets, same 30,000 samples, and we consistently process it through one single pipeline, harmonize the metadata, which is what we mean by harmonization to begin with. And then, we have a very favorable apples to apples comparison, the most reasonable comparison that one can do with these three corpus: data at source, reads, and polyharmonized. So this was the setup, this was the background that we of the experimental design that we chose to do this benchmarking study. So moving on, I also wanted to spend a couple of minutes on just the anatomy of the questions, right? So here on the right, you're seeing a table. This is a snapshot of any tabular data that comes out from an omics experiment. In this case, it's a RNA-seq experiment. The numbers are gene expression. Rows are genes or transcripts in this case. And the columns are different sample of the experiment with the corresponding metadata. And here you also have the metadata of the experiment itself, which you can capture from geo abstracts or the corresponding paper in cases where it's available. Now, if this is the anatomy of the table, we define 3 levels of questions. Number 1 are queries that you can ask based on the metadata of the dataset level only, right? So this is what we'd call level 1. Just to give you an example, I would ask, I'm looking for dataset that give me knockout of gene X studied in this tissue, right? Often that level of information is captured in the metadata of the dataset alone itself. Level 2 is a little deeper, where we are using the metadata of the samples. And a query, for example, would look like find datasets where the effect of some drug related to untreated control was profiled in some tissue that you like, right? That's level 2. And the third level, which is the most rigorous, is where you start asking questions about the gene expression level itself. Like, give me datasets where your favorite gene is more expressed than some other control gene, right? So this is the anatomy, and this is the systematic study that we did, and combined cumulative, like we asked 450 such questions, and we have published in the white paper all those questions, so you can, and the corresponding data sets as well, so you can give it a shot on your own laptop, desktop, and see what you learn. Um, there are three level of questions that we'll ask the model for all the three data sets, the corpus of data set that I described earlier. Now, in all of this, it's important to, again, establish the ground truth. It may be not perfect, but it is important to establish that. And we use the same, um, you know, trick that we used in the last study, and also, was used in many papers, where we had um, human experts, bioinformatics scientists, who asked the same 450 queries manually on these three corpus, right? And then that establishes the ground truth for this particular study. Uh, it's not engaging. I wish I was, being able to answer questions in real time, but I'll continue with my slides in the interest of time, and I hope I get to questions at the end. So, here's what we started finding. So I'll give you example of just one query. This is one out of the 450 queries that we ran. And in this case, I asked, hey, fetch studies that compare gene expression profiles in dorsal root ganglion. And, I got 6 false positives from Geo. I got 2 false positives from Creeds. And Poly, which gave me all the 3 data sets, which are 100% aligned with what we are calling the ground truth, where human experts, bioinformatics scientists, asked the same query to Geo and found these 3 data sets to match to answer the query. Now, to evaluate the performance of the models, and we are using 2 metrics. One is aggregated ranking, and also the F1 score, right? So it's not F1 captures both precision and recall, where you want to make sure that whatever results you're getting is accurate. And the second part is you want to make sure that you get all the right results that you want, right? So in this case, F1 is 1 because you get all the 3 that are supposed to come, and only 3, and there's no other data set that comes up here, right? And they're also ranked 1 in terms of relevance. So that is the performance of this harmonized data set is as good as it can be. Not so impressive for Geo. Now, this is just one query, right? We ran this for 450 queries for both level 1 and level 2. And here's what we saw. Consistently, polyharmonized data performed very, very well in this context for these 30,000 samples. Data at source was, modest at best, and Keats, outperformed it in some cases. So this again, perhaps the whole thesis is not super controversial for a lot of people, where we are arguing essentially that high quality data, harmonized data, would lead to better performance, and we can all agree and nod our heads. But to our knowledge, this is the most systematic and comprehensive study that we are planning to put out. We have already put this out as a white paper, but this again establishes and quantifies as to how to harmonize and what does that lead to. And hopefully this can, generate more conversations around these topics. Now, moving on to the level 3 problem, which, if you recall, was based on the data matrix. So in this case, for just to illustrate an example, the query is something like this. Find all RNA-seq samples where the gene expression value of my favorite gene is greater than that of a control gene. Now, at source, this will not give you any output because Geo does not have process data in all the cases. Even if it is processed, it's not in the same form, it's in processed through different pipelines and so on and so forth. The heterogeneity and the challenges of that are pretty well established and documented. We have been talking about that for a long time. So I'll not go into that, but the contrast is quite dramatic, where such queries just cannot be run on data at source or even at Creeds. But since we have harmonized that, meaning all these 30,000 samples have been pushed through and processed through one single pipeline every single time, we have the power, we have the luxury of being able to answer and respond to these queries pretty much on the fly. And so when you ask this question to polyharmonized data, the corpus, the third corpus, you get a plot like this, which you can interact with. The corresponding table is available as well. You know which data sets and samples are these coming from, etc., etc. Right? And of course, one can dig deeper into this. So if I were to summarize, right, the benchmark study that we did, level 1, level 2, we show clear quantitative improvement because of harmonized data in a very systematic way. And we are very happy to put this out in public domain. The third level of query, which I think is very valuable as well, is just not possible if you do not have harmonized data. So again, the value of getting data harmonized, depends on the context. In some cases, it's quite dramatic, where it just would not enable you to answer some questions. In some cases, the value is more incremental. Irrespective, qualitatively, we have seen that in a lot of use cases that we have used, it has always helped. The extent of how much it has helped is varies, but it always helps to harmonize data and then ask such questions. So moving on to some more real life applications, not as controlled as what we had in this benchmark study. We are trying to uh, classify different classes of problems that benefit from having high quality harmonized data that feed into such models. So one use case that we have seen, and I'll get into some detail, not a lot of it, is around information retrieval, where again the use case is quite similar and very real. As a bioinformatics scientist myself in my last company, often my colleagues would come and ask me, "Hey, is this gene or is this a line a good model for these gene expression, or what is the gene dependency of in this cell line for this particular gene that I care about this week?" And often, those questions I would either answer that the next day, or sometimes never, because I was busy with other things. But this is not a unique experience. Like most of the bioinformatics scientists, if not all, share this experience. This, and we are trying to see if technologies like LLM built on top of high quality harmonized data can address such concerns and scale up the bioinformatics team without compromising their colleagues, where they can get access to high quality information, retrieve it quite easily using just natural language. The second use case that we are seeing is there's a broad interest to fine tune the LLMs for specific tasks. Again, I referred to this in the first half of my presentation as well. But we are seeing some specific examples from at least a few companies um, uh, th- that are taking models, taking huge corpus of data, different types of data, and fine tuning it to perform a particular task. There's some that are already in public domain, like single cell GPT, gene PT, and increasingly, like you'll see more of such papers come out almost on a weekly, monthly basis. So there are two case studies that I'll allude to. For a variety of reasons, I cannot get into a lot of details here, but again, happy to have a conversation around that, around information retrieval, which in this scenario will imagine a small biotech company that is exploring potential targets in KRAS, dependent lines across different indications from many sources. Again, this is a very typical problem. You can replace KRAS with your favorite gene, but the problem statement remains the same, definitely in oncology and also in other therapeutic areas. So this is mostly to give context, not a whole lot of detail, but you can imagine how this can help in other places too. Now, to be able to enable this, right, harmonizing data is one part of it. So I wanted to give a sense of the architecture of the infrastructure that is needed again for the engineers and the research informatics scientists and computational biologists in this audience too. This would be of interest, where I can suggest or highlight different pieces of this architecture diagram. And at the bottom, you see data sources, in-house data, and your usual suspects from public domain: DepMap, GTex, TCGA, HCA. You can expand that quite easily and nicely. Irrespective, different types of data come from different sources. They're often in different formats, and they need to be harmonized through data models, connectors, and there's a whole tech stack around this, which is what we have built and uh, in a very differentiated way over many years. Once the data is harmonized, you also need to put this in some kind of a retrieval system, which allows LLMs to interact with it in a fast, efficient way. And then you have very specific tools to plot the data or to get the context of target ranking, gene expression analysis, or admit properties and whatnot. These tools, this repertoire of tools is essential for and that makes up your reasoning engine, right? Where plain natural language queries can be translated into a very context specific um, query to be able to give you right answers, not just any answer. So this is the architecture. I'll not go through this GIF, but what all of this led into is this is a representative screenshot of how you can ask queries in plain English to answer questions that are very typical about give me a snapshot of total number of shared hotspot mutations in a cell line which have a chrono score of greater than -1, right? To be able to do that, typically you would go to DepMap, cBioportal, and a variety of other tools, and you would get that. But a lot of colleagues would not be as familiar with cBioportal and DepMap, and they would rely on their bioinformatics colleagues to help them with this. We are imagining, like with tools like this, which are built on top of harmonized data from many sources, such journeys can be quite enabled. This also refers to the third, part of what we are imagining to become more mainstream is that the consumption journey, in this case, just retrieving information from a variety of sources, very complex data sets, with large dimension, becomes enabled by writing queries in plain English. You don't need to know SQL. You don't need to know. You just need to know your biology, and to evaluate um, the answers that you get. You need to make sure that that's not hallucinating, as it sometimes does. But with tools like this, we believe information retrieval within an enterprise will become more seamless and easier. And that's a very good example, a real benefit, real valuable use case that can be enabled by LLMs, which relies heavily on, in this case, as we demonstrated, high quality data. The other case, and after this, I'll stop and open to respond to questions. Is again, we are seeing this across different companies. Uh, this is just to illustrate and give some context. A biotech company wants to develop a foundation model from using large volumes of multimodal data, single cell, RWE, bulk RNA-seq, imaging data as well, and mostly from human samples. You can imagine um, a foundation model built on cell line data as well. So there are a variety of versions of this question, but in an abstract sense, the fundamentals remain the same. And what we are doing in this case is to um, take data from public sources in large volumes, and also patient data from different partners, RWE, response data, and so on and so forth, and push it through our harmonization engine and get it MLAD, which can now be fed into the foundational model ecosystem, which is quite complex and nuanced. We have, learned a lot working on this for the last many months. But the first piece is the data that is MLAD that feeds into this. In this case, we are sharing some screenshot of how we have been able to identify around 12 million human samples. And mind you, this is across different therapeutic areas and also different modalities of data, mostly in public sources. These are not cell line data; these are human sample data. Um, and we have the capability to ramp up our harmonization engine by 4x in this case. And we love such challenges where our skills or capability of what we can do on a monthly, weekly basis can be pushed to the limit. That's where I think we can innovate and create a lot of value. But this is one exciting example where we may have to ramp it up and more to provide 12 million harmonized data sets to feed into a foundation model. And then once you have the data which is ready, that feeds into a whole layer of infrastructure which has many different components, um, which again, one, I'm not super qualified to get into a lot of details here, and this forum is also not quite ideal for that. We'd be happy to respond to any questions outside of this. Now, this foundation model, this exercise, this project is it's in very early days. It's not built yet. It's not clear as to what questions will it answer and how impactful will it be. But we are very excited and we believe in this promise. So in the next hopefully 6 to 18 months, this picture will become more clear. But what is undeniable and unmistakable is that there's a critical mass of companies who are very keen and are defining and designing and building these foundation models. And almost all of them would require high volumes of data, some of that which would be very, very highly harmonized. Otherwise, you'd compromise the otherwise it would be your any such foundation model initiative would be at risk. So in this case, we're hoping we are seeing some early signs of where we have been able to give faster access to large volumes of multimodal data compared to if you were to acquire it yourself by scraping and going to different portals and so on and so forth. Having a grade of quality as I captured in that pyramid is important and highly non-trivial. We are hoping to show that it translates into successful and more accurate foundation models as well. And finally, you save time and money, but the what is far more exciting is that how these foundation models will impact um, the R&D journey of small biotechs and large pharma companies combined. We are very keen and looking forward to partnering with such groups to enable them. Um, but again, stepping back, right? Like I wanted to acknowledge that there's a lot of interest in this field, and for good reasons. We are seeing some very clear business case that are being addressed by these technologies. There's also a lot of hype, and there's also a lot of hyperbole, which is inevitable. Like it goes with the territory. But as practitioners, right, our understanding, our approach is evolving on a daily, weekly basis. And as must be true for others who are trying to deploy these in their own settings, right? So our intent to have this webinar was to facilitate that conversation, to join that conversation, to invite uh, folks who are on the fence, and just enable a broader adoption, because it is a promising tool and technology at the end of the day, right? And we think we can leverage that more to help with some very fundamental efficiency and questions in R&D in life sciences and pharma. With that, I'll stop and I'll thank Zahid again, Pistoia, and all of you for participating. And I'll be open to questions.
Wonderful. Thank you, AJ, for an excellent presentation. We have had a few questions in the Q&A box, and maybe we can start with those. So firstly, from Tugrul, you mentioned using other LLMs in addition to BERT. Would you mind sharing which?
Yeah, so in the case, in the benchmark that that I shared, we used GPT-4. And our focus was to highlight just one variable that was different, which is the harmonized data. So we did not want to introduce another layer of complexity by including Llama and others. So for that, we use only GPT-4. We have played with other models as well, like Mistral, Llama, and such. But in our production pipelines, we rely almost exclusively on BERT and GPT-4. Depending on the task, like they give you different kind of performance.
Thank you. And a question here from Francis. It looks like there are two questions. So how structured, curated does the data need to be for these models? Should the objective not be to use this technology to move away from the need for expert curated data? And then then a follow-up question to that: when you speak about data quality, what do you mean?
Right. So there are a bunch of questions, so I'll try to respond
Yeah.
to one. And Zahid, if I miss one of them, let me mind me. So what does quality mean? I'll start with the last one. Um, that's a very difficult question, but we have taken a stance. So in this case, we have a list of around 50 QA QC checks. Some that would fall into what I would call a experiment design question, right? How many samples are there for a given cohort? Is that less than three? We would capture that. We would put that out. There's some questions around the analytical part of the workflow, right? Which reference genome was used? So there are, 50 such detailed QA/QC steps that we publish as a QA/QC report. We call it poly-verified reports for each data set: bulk RNA-seq, single cell, microarray, proteomics, metabolomics, right? Now, is there a agreed upon list of these things? No, there's not, right? But I think, as community of scientists and engineers, right, we often hesitate from drawing a line because these things are ambiguous to begin with, but we can all agree on at least what is a uh, else least common multiplier of sorts, right? Like where all of us agree that this is the list of QA/QC metrics that each data set should go through. Now, we have the advantage of working with many companies and supporting them with their data quality needs. So our quality metrics also evolve and grow. I'm saying that today we have 50 QA/QC metrics. Next time when I'm doing a webinar, I may say there are 75, right? Because we keep adding, so we are growing. It's not perfect, but it's very actionable. So you can decide in less than 5 minutes if it's worth spending another 30 minutes on or not, if you evaluate that QA/QC report. So that's one. The second question, if I remember right, is that how harmonized or how high quality does it need to be, right? Um, so one of the messages that I want to send out very clearly that the answer is not very satisfying, but it depends, right? So it depends on the use case. In some cases, like you can go to Geo and search for data sets or Google, right? You can do that. You will get some answers. I don't know how effective and how meaningful they are, right? Definitely on Google, Google does a pretty bad job of finding omics data sets. Geo does marginally better, but not a whole lot, right? But if you were to find queries like level 3 that I was talking about, you just cannot do that on Geo, right? For there, you are, critically bound to have harmonized data, right? But if you want to do a simple query just on the metadata of the data set, Geo may give you some answers. It may not be perfect, it may not be great, but it will give you some answers. Having harmonized metadata of the data set level will improve that performance as well. So what we have seen almost in every single case, it does help to have harmonized data. Now, to what extent does it help? Depends on the use case. And in some cases, like when I'm asking questions on the data matrix, it's absolutely critical to have it. In some, not so much. Now, if you're building a foundational model, right? As I try to capture in the pyramid, you don't need the whole corpus of everything that goes into foundation model to be super high quality, right? But having the most top layer, which is most targeted, most specific to the task that is going to perform, I think that will increase performance. We have shown that in some cases we'll do that more. And finally, is the promise not to move away from or take advantage of unstructured? I think that has happened. That will happen more. There's a paper that came out, Gene PT, right, which takes just the text data, right, which is not as structured, and feeds into that to ask questions about genetic signature. I believe we're not at a place today. The state of the art is not at a place today which enables you to take advantage of unstructured data as you'd like to. Maybe 3 years from now, 5 years from now, I mean, clearly that direction will move. But even for large language models, right? There are papers which are not in life sciences, right? Just in the large language model space, where folks have shown that smaller models which have access to high quality data outperform bigger models, right? And why is that relevant? Because building and running and deploying large language models is a very expensive exercise, right? It requires a lot of people. It requires a lot of computational power. So if you have access to high quality data, you can outperform such big models too, right? So um, I hope that commentary and touches upon some of those things.
Yes.
But a lot of these things are evolving as we speak as well, right?
Thanks, Adrian. And Francis, I hope that answers your questions. So a question here from Avani. The data quality will be different for different data omics. How is then the quality metrics managed and measured?
So in a lot of the um, construct of the quality reports that we publish for different types of data have a similar theme, right? They would be about experimental design. They would be about the analytical workflow that was used to process the data, and they would be about the metadata, right? The details would be different for proteomics data versus single cell data. But we're trying to capture that much like what a scientist would use to evaluate if she were looking at a data set to see if it's worth spending her time or not, right? We try not to be the judge, but we try to just expose the metadata, right? So that you can make the call that are 3 samples per cohort good enough or not? Maybe in your case it is, if you're just testing out a hypothesis. Maybe it's not if you're building a deeper statistical model, right? Maybe that reference genome is good. Maybe Callisto pipeline is good enough. You don't care about isoforms. Maybe it's a deal breaker if you want information about isoforms. So we are trying not to judge, but to expose those parameters so that you can evaluate very quickly if it's worth spending another, half an hour of your time or your team's time or not, right? So in an abstract sense, we are following the same structure for all the quality reports for all data types, but the details of those are different as they should be.
And this is an anonymous question, but are there any foundational models available for pharmacovigilance?
Not that I can recall from the top of my head, but if I were to guess, I would say most likely yes. It would be a matter of doing some PubMed and Google search.
Mhm.
I don't know how good they would be, but I'm sure they would be.
Yes.
Yeah.
Uh, and then a question here from Clarence. In the life sciences domain, does the high level of noise in large scale studies contribute to higher chance of hallucination in LLM output?
Yeah, so hallucinations again is a very widely discussed topic. In some ways, I think it has helped that almost everyone is aware of hallucinations, right? Now, if you're writing, a poem or something funny in ChatGPT, if it hallucinates, you can have a chuckle. But for life sciences, where a lot of the questions are quite critical, right, and any follow-up experiment would be quite expensive, hallucinations could have a real impact. So what we see again in some use cases that having harmonized data reduces the chance of eliminates hallucination. Does not eliminate it; it reduces the chance. But I think if you learn something from LLM models like the query interface that I was showing you earlier, right?
Yeah.
Often any such hypothesis will be discussed and debated as it should be. So there is an element of hallucination which cannot be completely ruled out, right? The way Gen AI or LLMs models are designed, it is inevitable, right? But there's also a number of activities outside of Elucidata. In fact, in the broader tech space, right, Gen AI space, where people are publishing methods and models to reduce hallucinations, not to eliminate it. That still seems to be beyond reach right now.
Thank you, AJ. And then we have a question here from Hans. NER is a form of structured data extraction from narrative sources. Do you propose to harmonize narrative text?
What is narrative text like?
I think if you remember when you were your I think it was your second or third slide.
Yeah.
Yeah.
And I also wanted to make a comment about hallucination at large, right? Like, um, I don't mean to be tongue in cheek, but like, as an industry, the failure rates that we have in our, phase one, phase two, phase three, the hallucination rates are much smaller than that, right? There are a lot more checks and balances built around hallucinations in LLM models than some far more material changes, right? So as a community, I think we should be okay to embrace this nuance of a tool, right? Any tool has such limitations. This one comes with this.
Okay. Perhaps Hans, we can follow up after the call, after this session, if you if that is what you need. But there's another question we hear from Deb. Can you provide more information about the harmonization tech stack?
Yeah, I can. Some, right? So there are three pieces to it. Number one is connectors. These connectors take data from raw form that comes out of the instrument. That's a fast QC, and turns that into a table that you can read gene counts. Or that's one piece. The second piece is the metadata harmonization, right? Where the sample metadata or the data set metadata or gene IDs that is harmonized. And for that, we pull data from your ELN or from geo abstracts or PubMed paper. Sometimes you find that in geo abstracts, sometimes you have to dig deeper into the supplement information. So we pull all of that. That's the second piece. And the third piece is publishing a QA/QC report. So where we give you a very granular picture of all that happened with your data. Like for example, what reference genome was used, what metadata ontologies were used for disease mesh or ICD-10. We expose all of that for you to evaluate in less than 5 minutes if it's worth spending another half an hour on this data set or not. So those are the three steps of our harmonization engine. Again, perhaps a more detailed conversation is warranted if you want to dig deeper into proteomics or single cell or spatial transcriptomics, etc., etc.
Thanks, AJ. Okay, so I think we've reached the top of the hour. What I'd like to do is to thank AJ for his excellent presentation and for fielding some great questions that we've had in the last sort of 20 minutes or so. There have been a few questions that we haven't been able to get to, and I apologize for that. But I will be sharing this with our colleagues at Elucidata, and perhaps they'll be able to get back to you separately.
Zahid, if you may, if you want to share those questions with us outside this,
Yes,
we'll respond to you and you can share the response with the rest.
absolutely. Absolutely. So thank you again, AJ. Thank you